We have mentioned DNA methylation before, but we have yet to go into detail about what's going on. So DNA methylation usually refers to a methyl group on cytosine. So here we have a cytosine, and DNA methyltransferase is responsible for putting a methyl group on this cytosine at the 5 position, creating a 5-methyl cytosine. Now, remember, DNA is a double-stranded molecule, and that cytosine binds to guanine. So you can have an unmethylated DNA, a hemimethylated where only one strand is methylated, or fully methylated where there's a methyl group on both strands in that area. So how does methylation affect transcription? There are 2 ways that it can do it. One is, so let me back up again. So usually around the promoter in eukaryotes, you have these CpG islands. And the CpG islands are cytosine and guanines, okay? So you have this cytosine and guanine area, and that area is subjected to methylation. Now, usually, remember we talked about constitutive genes, so those housekeeping genes that we need on a lot, they tend to be unmethylated, whereas tissue-specific genes are going to be methylated in some and unmethylated in others, depending on which tissues need it. So in this way, methylation affects transcription and allows us to be specific. So it does it in 2 ways. The one way is it's like this physical blockage to stop the activator from coming on. So if you have all these methyl groups in that area, it's almost like shielding it, okay? So the activator, it can't get in and bind. It can't even see where it's supposed to go. So this is a physical blockage. The other way is when you have these CpG islands that are methylated, they can attract methyl CpG binding proteins. Now those proteins now can come in and recruit other proteins that can cause this chromatin to revert to a closed formation, okay? So methylation can physically block the activator proteins from getting into the enhancer, or it can recruit proteins that change the chromatin structure. So that's 2 ways that methylation can inhibit transcription, okay? Methylation tends to inhibit transcription. It's also heritable. What does that mean? You can pass it on, okay? So think too like with epigenetics. So you can pass on these sequences that are methylated. You can tell the cells that, this cell needs to be methylated. So how does that happen? There is a de novo methylation where a cell can be newly methylated, and then there's maintenance methylation where we're going to preserve where methylation should be. Okay, so we can pass on this methylation by this maintenance methylation. Now, we talked about epigenetics before, so I'm not going to belabor the point here, but understand that epigenetics is a way that we can affect transcription. So we have talked about like X inactivation, where you can't transcribe the genes on X. We talked about imprinting, where you can only transcribe genes depending on which parent it comes from. We talked about that this is possible through methylation and through chromatin remodeling. Well, now we went into further detail to allow you to connect the dots of how epigenetics works. So I just want you to bear in mind that these modifications can be epigenetic related. So you can have this methyl group. And remember, epigenetics is being passed on. So a change in expression, but not a change in sequence. And so DNA methylation, so having this maintenance methylation, you can inhibit genes by chromosome remodeling, and you can, pass on how those chromatins should be remodeling by putting on different modifications or variants or having those feedback loops. So these are all ways in which we can have epigenetic control where we change the gene expression and we pass that on. And another way is these non-coding RNAs that we talked about, okay? So non-coding RNAs can play a role in our epigenetics and in our transcription. So most of what we talked about has been how to affect the gene expression at the transcriptional level. Now we're going to briefly talk about translation and RNA stability. So if you think back to bacteria, the same thing. If you can't get to it, you can't access it, the ribosome can't access it, it can't transcribe it. And there are ways in which eukaryotic mRNA is made stable, like by adding a cap and a tail. Well, if you don't have a stable RNA molecule, then you can't transcribe it. So to consider this, we're going to look at how iron gets into our cell. So iron is regulated by RNA binding proteins that affects translation as well as the mRNA stability. So when we ingest iron, so here's our iron, right? It binds to this transferrin. This transferrin binds to a transferrin receptor, this endocytosis, so it takes the whole thing inside. Once it's inside, that iron is released, and then it can go bind to proteins that require iron, and if we have extra, it can be stored in this ferritin. Well, we have to have a way to create transferrin and ferritin. So for that reason, we have this, here is the ferritin, and here is transferrin regulation, okay? So here we can regulate at the transferrin by looking at this IRP, so this iron regulatory protein and this iron response element. And so they work to help us either produce what we need to assimilate the iron or not produce it. So this ferrin has this iron responsive element that sits here upstream the start site, and this is the 5-UTR or untranslated region. And so if this IRP binds to the IRE, okay, so there's a mouthful. So IRP binds to the IRE. So when that happens, it inhibits translation, okay? So if these things are bound up, it's going to inhibit translation. Our ribosome can't attach and scan, okay? And so if a ribosome can't attach and scan, we can't translate it. Well, another thing that can happen, so remember, these can exist in two forms. So if we have a lot of iron, then our iron binds to our IRP. If the iron binds to the IRP and changes its shape, it can't bind to the IRE. If it's not on the IRE, then our ribosome can interact with this area, scan it, and produce the ferritin protein. All right. Now, a similar way we can regulate the transferrin receptor. So here we have our mRNA, we have our start codon. Well, our IRE is downstream here, okay? So it's now in the 3' UTR or untranslated region. So now what happens, lost where I was. So now what happens is it binds here and helps this to be a more stable molecule. Okay, so by this stabilizing the mRNA, it's allowing translation to occur. Whereas here, if we have an abundance of iron, the iron binds to the IRP can't bind to the IRE, and now we have endonucleases that's going to degrade it. Okay, so for ferritin, we have the IRE upstream, and the IRP binds to it and inhibits translation by blocking the ribosome. Or if we have an abundance, then we have the iron binds to the IRP, takes it off, and allows translation to occur. With transferrin, it's going to help with stability. So by the IRP binding, it's going to stabilize this molecule and allow translation to occur. If it's not bound, then the endonuclease comes and degrades it, and it's not stable. Okay, so these are additional ways in which we can affect gene expression in eukaryotes.